السلام عليكم ورحمة الله وبركاته. رجعنا لكم مرة تانية مع الميكروبيولوجي MCQ for laboratory scientists and technologists for license exam. أسئلة الامتحان بتاعت الميكروبيولوجي للحصول على ترخيص مزاولة مهنة التحاليل الطبية ده part 1. Please subscribe, like and follow to see the part 2 which is coming soon. We'll start with first question. What is the significance count of bacteria colony forming unit per ml? 100, 1000, 10,000, 100,000. The correct answer is 100,000, more than 10 to the power 5, which will be heavy gross. So the correct answer is 100,000. Which of the following is not Romanovsky stain? Gram stain, James stain, Leishman stain. The correct answer is Gram stain. Romanovsky stain consists of eosine plus alkaline methylene blue, eosine only, methylene blue only, Indiana ink. The correct answer is eosine alkaline methylene blue. Difference between gram-positive and gram-negative bacteria is cell membrane, cell wall, nucleus, cytoplasm. It is the cell wall. The difference between gram-positive and gram-negative, it is the cell wall. Secondary stain of gram stain: crystal violet, iodine, safranine, malachite green. The correct answer is safranine. The primary stain of gram stain: crystal violet, iodine, safranine, malachite green. The correct answer is crystal violet. Gram-negative bacteria color appear under microscope: dark purple, pale to dark red, orange, blue. The correct answer is pale to dark red. Gram-positive bacteria color appear pale to dark red, orange, blue or violet. Correct answer is blue or violet. In gram-positive bacteria, there is peptidoglycan and teichoic acid, has no peptidoglycan, has no teichoic acid, none of the above. The gram-positive bacteria have peptidoglycan and teichoic acid. In gram stain, which of these is used as a basic? Methylene, methyl violet, crystal violet, safranine, neutral red, gram iodine, acetone, alcohol. The correct answer is methyl violet, crystal violet, which is a basic stain. On gram staining, one of the following bacteria form chain: streptococci, staphylococci, mycobacterium, diplococci. The correct answer is streptococci, which appear in chain. It will be like this. Staph is a grab-like. It will be like a grab-like forming. Diplococci, it will be in two cocci attached each other. Gram-positive cocci arranged in groups, clusters. These gram-positive cocci which are arranged in groups will be streptococci, staphylococci, cocci, spiral. As we mentioned before, streptococci looks like this long chain cocci. The correct answer will be staphylococci. Which of the following microscope would you use to examine a smear of bacteria stained by gram stain? Electron microscope, dark field microscope, fluorescent microscope, bright field microscope. The one will be used to examine a smear of bacteria will be bright field microscope. It is light microscope which commonly used in laboratory. Gram-negative bacterial cell wall characterized by the presence of large amount of glucose, large amount of glycogen, large amount of cellulose, toxic lipopolysaccharide molecule. The correct answer, toxic lipopolysaccharide molecule. Lead agar used in urine culture because it is used as a transport media, it is basic media, help colonies count, and prevent swarming of Proteus. All of the above. The correct answer is help in colonies count and prevent swarming of Proteus. Tire-Martin media is a choice of the isolation of the following organism: Pseudomonas aeruginosa, Haemophilus influenza, Neisseria gonorrhea. The correct answer is Neisseria gonorrhea. Media used for tuberculosis: Lewis Jensen media, nutrient agar, urea bros. The correct answer is Lewis Jensen media. Bacteria can grow in the presence or absence of oxygen are called anaerobic facultative, anaerobic strict, anaerobic none of the above. Anaerobes, tis, and strict anaerobes in the absence of oxygen. So if bacteria grow in the presence or absence of oxygen, it's called anaerobic facultative. So the correct answer is facultative anaerobes. Optimum temperature for the growth of bacteria is 50 degrees centigrade, 60 degrees centigrade, 37 degrees centigrade, none the above. The correct answer is 37 degrees centigrade, which is the human body temperature. Gas gangrene caused by Clostridium preferens, Clostridium tetani, Clostridium botulinum, none of the above. The correct answer is Clostridium preferens. In UTI, urinary tract infection, the following is present: glucose, acetone, leukocyte, none of the above. In urinary tract infection, you will find, too many white blood cells, leukocyte as indication of infection. A child diagnosis shows scarlet fever. The cause is Streptococcus pyogenes, Staphylococcus aureus, Bacillus anthracis, Pseudomonas aeruginosa. Scarlet fever is caused by Staphylococcus pyogenes. Test for identifying strep pneumonia: catalase, oxidase, or optician. The correct answer is optician. Confirmatory test used for Streptococcus pneumonia: catalase, optician disc, coagulase, bile solubility. The correct answer is bile solubility, used as a confirmatory test for Streptococcus pneumonia. Media used for the detection of hemolysis is blood agar, neutral agar, McConkey, all the above. The media used for the detection of hemolysis is blood agar, B alpha or beta hemolytic. Most common bacteria that produce urease are Proteus, Pseudomonas, E. coli, none the above. Proteus is known that is one of the common bacteria producing urease. Substance used in catalase reaction: hydrogen peroxide or carbon dioxide, H2O, HCO3. The substance used in catalase reaction is hydrogen peroxide. To help in identification of Pseudomonas and Neisseria, this test is citrate test, coagulase test, indole test, oxidase test. The correct answer is oxidase test. Pseudomonas and Neisseria known are positive oxidase. A specific test used for Staphylococcus is catalase test, coagulase test, optician sensitivity test, CAMP test. The correct answer is coagulase test. Which of the following bacteria need a dark field microscope to detect? Treponema pallidum, Klebsiella, Streptococcus pyogenes, Clostridium species. The correct answer is Treponema pallidum, because it is a spiral bacteria, so you will see it better under dark field microscope. Which of the following is strict aerobes bacteria? Mycobacterium, Klebsiella, Streptococcus pyogenes, Clostridia species. The correct answer is Mycobacterium, which like Mycobacterium tuberculosis. Which of the following is capsulated bacteria? Mycobacterium, Klebsiella, Streptococcus pyogenes, Clostridia species. The correct answer is Klebsiella species is capsulated with mycoid capsule. Which of the following media used for differentiate between lactose fermenter and non-lactose fermenter? McConkey agar, chocolate, blood agar, XLD. The media used for differentiation between lactose fermenter and non-lactose fermenter is McConkey agar. The MVIC test reaction of E. coli is plus plus minus minus, minus minus plus plus, plus 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 minus, minus plus minus minus. The MVIC test is for I for indole test, M for methyl red, V for Voges Proskauer test, C for citrate. These are chemical tests used for identification of E. coli and the other Enterobacteriaceae family. This reaction for E. coli is indole positive, methyl red positive, but Voges Proskauer and citrate both are negative. So the correct answer is first choice. All the following are prokaryotic cell except fungi, bacteria, chlamydia, mycoplasma. All are prokaryotic except fungi is the correct answer. The obligate anaerobes bacteria will grow only in the presence of oxygen, grow only in absence of oxygen, grow either in presence or absence of oxygen, grow in presence of oxygen and traces of 5 to 10% of carbon dioxide. Anaerobes will grow only in the absence of oxygen. So the correct answer, absence of oxygen. But presence of oxygen called aerobes. For in the presence and absence of oxygen, this is called facultative anaerobes. Which need oxygen and carbon dioxide, this called fastidious organism, like Haemophilus. What is the selective media for fungi? Blood agar, McConkey, nutrient agar, saprolid dextrose agar. The correct answer is saprolid dextrose agar. Bacteria which don't cause hemolysis is beta hemolytic bacteria, alpha hemolytic bacteria, gamma hemolytic bacteria. Which bacteria which don't cause hemolysis called gamma hemolytic. Bacteria without cell wall is chlamydia, rickettsia, mycoplasma, spirochetes. The correct answer is mycoplasma. We came to the end of part 1 of microbiology MCQ. Please share, subscribe and follow to get the part 2. Assalamu alaikum wa rahmatullahi wa barakatuh.